Good afternoon, everybody, and welcome to the final session of this second day, which is about gastrointestinal viruses. My name is Lasse Rasmussen, and I'm from Statens Serum Institute here in Copenhagen. And I give the word to my co-chairman for introducing the keynote.
Good afternoon. So our next keynote speaker doesn't need very much presentation. First, because he is a well-known expert on enteric viruses. And secondly, because he has always been a very loyal participant at our ESCV annual meetings. Dr. Jan Vinje is the chief of the National Calicivirus Laboratory and director of CaliciNet at the CDC, at the Division of Infectious Diseases. He has published about 200 papers on enteric viruses, mainly on noroviruses. The last one on an update classification of genogroup and genotypes in noroviruses. Please, Jan. The title of his lecture will be Progress and Challenges in Understanding Norovirus Infections.
Okay. Thank you very much for the introduction, and thank you very much for the organization for inviting me. What I've put together is an overview where I changed a few slides because Dr. Goodfellow yesterday presented some of that work, so I didn't want to have too much overlap. So I tried to give an overview on in the next slide on the left. And for those people that have never heard about norovirus, you have some news bites. The one left under the oysters, I like that one the best. The possible chink in the armor of Satan's bioweapon. It can only be a UK tabloid, of course. But yeah, so norovirus is a virus that is causing acute gastroenteritis, and thanks to the improved diagnostics over the last 10 years, I think we have much more information on its burden. And also, there are 2 vaccines currently in the pipeline and clinical trials, at least 2. I think in China there are also 2 in clinical trials. So it's an important virus to study. And I will go over the burden of disease, the virus classification, and diagnostic methods, how we use them in surveillance, and give you some of the data that we have achieved from those surveillance systems, both for outbreaks and sporadic cases, primarily in pediatric populations. And now we're expanding that also to a more global surveillance in the light of the upcoming vaccines, knowing exactly what strains are circulating. And then I will end with an update on where we stand with the vaccines that are currently in the pipeline. So the clinical disease is pretty straightforward, and as Dr. Goodfellow Ian yesterday mentioned, it can be pretty horrible, not only in Florida, but almost everywhere, because the virus infects everybody globally. For most people with a healthy immune system, it's just a miserable couple of days, but there are people that are immune compromised, or the elderly, and I'm talking about the elderly, traditionally was like 65 years and over, but it is now more like 80 years and over, primarily in the nursing homes and long-term care facilities, that are very vulnerable for these infections. In the developing countries, the children are mostly vulnerable, and the burden of disease of this virus is the highest among children younger than 5 years of age. There is a lot of asymptomatic shedding, and the reason that is important is that norovirus is also an important foodborne pathogen. It's causing more foodborne outbreaks than all the bacteria together, including the E. coli and Salmonella. And so foodborne workers, if they shed the virus asymptomatically, and they may prepare a salad for the salad bar, you can see how easily an outbreak can occur. Since the diagnostics was improved, a lot of information has been published, and that was sampled by the WHO Foodborne Disease Burden Epidemiology Reference Group. That's a long name for FERG. And they looked at what is the stratified, the age stratified, the estimates of disease and the deaths, and they looked at all foodborne hazard, and they found that norovirus was the number one cause of foodborne illness worldwide. And you see the numbers there. I don't go over it, but just to highlight that the estimates is that the virus is causing deaths primarily in children in the developing countries, about an estimated 200,000. What is also very important is not only the disease burden, it's also the economic burden. And there is a paper out based on this study from the FERG working group about the economic loss. And you can see it's up to $60 billion, that those are the estimates in societal cost. So it's an important virus, and then you look at these data. Obviously, you see differences if you look at the different continents, where the healthcare cost in the U.S., for example, is highest in the people over 55. That's basically those long-term care facility people that get hospitalized and have to stay in a hospital for sometimes up to 1 or 2 weeks with chronic diarrhea. So you see these differences. So this gives a very good information for regulators and also for scientists and for vaccine producers where to target. If a vaccine or an antiviral is developed, so where do you need to target? Which populations do you need to roll that out? In the US, in the last 10 years, there have been several studies conducted looking at the burden of disease at all levels of this pyramid that's here on the slide. And it's an estimate that is being refined, but the numbers are not much different than what they are shown here, which is about 20 million people are getting a norovirus illness every year in the US. And in particular, the hospitalizations is significant between around the 60 or 70,000 every year. There are deaths reported. You see the estimates there. Again, in the U.S., it's primarily in the elderly, in the long-term care facilities. And usually there's a comorbidity, so it's not necessarily norovirus is the ultimate cause of death. So, looking at the severe disease outcomes, you see on the left, you see the hospitalizations in yellow and the emergency department visits in blue. And you see that most of those, the highest rates is in the younger age group from 0 to 4 years of age. And then on the right, you see the more severe outcome, as I just mentioned. You see there's a little bit of red bar there from young kids, but it's very small compared to where most of the deaths in the US would occur, and that's in the over 65. So, and then you can look at different years if you have enough data, and this was shown in 2002, where you see an increase compared to the previous years. Then you see a dip to what we consider like a normal level of associated deaths. And then in 2006, another increase, and I don't have the data for the year since then, but it was typical that those increases of death rates were associated with years when a new norovirus strain emerged, and in a susceptible population. So you see there's an association with, in this case, typically there are G2.4 strains. This is the most prevalent viral strain. I will show you in a couple of minutes. So the virus belongs to the family of Calliciviridae, and as Dr. Weza just mentioned, that whole family has been officially now updated to 11 different genera. For humans, the most important ones are obviously norovirus and sapovirus, which is primarily a childhood diarrhea virus, here on the left also in green. And furthermore, there are some retroviruses that have been found in children. and it's here. It's primarily a virus that infects macaques, but there are a couple of individual samples found both in Latin America and in Bangladesh that are found from children. So the other viruses all have their different hosts. So the caliciviruses, every host has at least one norovirus that forms a very distinct cluster. There's no proof, no information, no data that supports that there is a zoonotic reservoir. So all these there is not like an animal reservoir where the virus can reside and then jump into humans. So it's primarily a strictly human virus, with some exceptions. And I will show you later. So the virus is a single-stranded positive strand RNA virus. Pretty straightforward, only 3 open reading frames. The 5' end, the non-structurals, where some of the studies with antivirals are targeted against the protease and RNA polymerase. But most of the information that we have is about the structural gene, the second open reading frame, the major structural protein, the VP1. And sequence analysis over the last almost 2 decades has indicated that there are a lot of recombinations occurring. And the recombination hotspot is in the center here, between the first and the second open reading frame. So traditionally, people have been sequencing either on the first open reading frame, that's where the first assays were developed like 25 years ago. And then typing occurs in the second open reading frame with codes for the capsid protein. But if you type only with one of those regions, then, and that's long time has been done by different groups, then you miss basically if a recombination occurs. And currently we have assays available that amplify over this junction region, and so a partial region of the polymerase and partial region of the capsid provides you with that dual typing information. The classification is based on these 2 regions. On the left, you see the VP1 typing, which can further be delineated into genogroups and under that genotypes, and then variants, in particular the G2/4s. And on the right, you see the polymerase, the RNA-dependent RNA polymerase typing. We there have P groups and P types. So we now have a dual typing system, which is similar as for rotavirus or for flu, for that matter, H1N1. We now have the P type and the capsid type, the C type. So we recognize 10 different genogroups that are also officially now confirmed in just recently published. And for humans, it still is that the viruses belonging to genogroup 1 and genogroup 2 causing most of the infections. On the right, you have the genogroup 1 with a prototype virus Norwalk. That's where it all started in Al Kapikian's lab in NIH in the early 1970s. But those viruses still cause a minority of the cases, both in outbreaks and in sporadic cases. Most of the viruses could be typed to genogroup 2, and of the genogroup 2, we now recognize 26 different types with G2/4 in the data that I will show you in the next couple of slides being prevalent and predominant for the last 15 years. There are some minor viruses that, this is an old G215 that belonged initially to the genogroup 2s, and now we can classify that as a separate genogroup. And there are some viruses here, genogroup 4. There is some indication that may have been a zoonotic event. Very rare, these viruses. They're still circulated, but they're also very close to viruses that are uniquely for dogs, pet dogs. So here is an overview of the genogroup 2 viruses. As I mentioned, there are many here in red that have been added in the last couple of, I would say, 2 years. They're primarily detected in rare pediatric reservoirs. For example, in Iquitos in Peru, where you only can fly and you cannot drive there. So this population there in northeastern Peru is pretty unique. And there, in collaboration with Johns Hopkins, we have been able to detect a couple of those rare viruses. But the question becomes, if these viruses could escape, they may become a pandemic. So at least now we have them in our table of types, and we can, with our assays that also detect these new strains, we're able to monitor if any of those will escape there and cause outbreaks and disease. So this G2-4s, they can further classify it in variants. We have seen variants with names. That is still the case. So we may have heard of those. The last one is Sydney, G2-4 Sydney in 2012. And before that, we have other names, New Orleans and Den Haag in the Netherlands. You see that almost every 3 years, that particular virus was replaced by a new variant. So it's kind of a genetic drift, like for flu. However, in 2015, we didn't see anything happening. It was still the Sydney variant that of the G2-4s. But then again, we're looking further, and we found that was a new recombinant. So the virus is still able to change itself, have a new polymerases, which may explain that the replication, the increased replication, and this virus is still circulating and causing about 50% of the outbreaks here in Europe, I would estimate. So looking at those recombinant strains here in color, so that new strain, that G2-4 Sydney, is here in blue. All the green ones are not typed. That's the time frame that there was only typing in the polymerase. I'm sorry, in the capsid. So these are all G2-4 Sydneys. But here in the blue, you see that this is the new variant, and we know that a lot of these here are from this older variant. So really, all in a sudden, we got this emergence of this new strain with this new polymerase. What does that mean? It's clearly a different epidemiology, and there are some other viruses have emerged in other places, and which I will show you in a minute. Other strains among those are this G2.2 in the middle. That is another virus that also had adapted the same polymerase. So this polymerase is able to, and it's a P16 polymerase, is able to recombine with a lot of different strains. That's what we have seen over the past couple of years. And that is summarized here, what's kind of the blueprint of what is currently circulating, and I put a red arrow there. That's P16, and you can see the numbers both from our databases at CDC as well as NCBI, the number of entries. And you see this P16 can recombine with almost 10 different capsids. So this is a very potent polymerase, and we still don't understand exactly what this polymerase is doing differently than the previous one. So the nomenclature, we also took the opportunity to, when we have all this increase in number of strains, to also update the nomenclature. And so on the left is how it used to be written in publications, and now we make it a little bit easier because we name first the capsid, which is the most important, and then the polymerase, and we put it between brackets to distinguish the new nomenclature from the old nomenclature. Otherwise, people would think that there maybe is a mess up and a mistake. So this is just published, and I would recommend if you're interested in this, then take a look at the publication, what the latest nomenclature is. So, and to help you with that, there are typing tools. The RVM typing tool here is I think has been there for almost 15 years. And at CDC, we also had a typing tool internally, but then we decided to also have it outward facing. And now we have exchanged the same reference sequences. Both typing tools should hopefully this week or next week give the same output if you enter sequence information there. So as for detection, most assays currently available are real-time PCR assays, but it has increasingly being replaced, at least in the US, in the hospitals, by the use of multi-enteric pathogen panels. And we will get, there are posters about some of those panels, the BD Max, I've seen a poster presentation. in the US is primarily the, and this is what the target region is for all these assays. It's the most conserved region between this first and second operating frame. A very robust assay has been developed basically in 2003, and some small indications, mutations in the primers over the years, but it's still very robust and very sensitive in detecting all these different norovirus strains. But the assay that is mostly used now in a lot of these laboratories is the BioFire film array. It's pretty expensive, but it is all in one pouch, and you don't have to do your extraction separately. So it's all in a pouch. You enter the sample, and in an hour you have results. Not only for the viruses, the five different viruses, norovirus G1, G2, and astroviruses, the enteric ones, not the ones that cause encephalitis, as we heard earlier this afternoon. And then enteric adenoviruses and sapoviruses. But there are others, and they're listed here. Some of them are FDA cleared, only FDA cleared for bacteria, but then usually the company would like to add on also validation studies for enteric viruses. So there's a tendency to move to these multi-enteric pathogen panels. And of course, whole genome sequencing, it's a little bit more challenging for these viruses because the stool has a lot of inhibition, and these enrichments are sometimes needed. For most NGS, sometimes it increases the sensitivity. You can do it with RNA baits. Or you can do that with simple filtration steps and getting rid of your bacterial populations if you're interested in this case, in enteric viruses. You can also do specific, and we have chosen for specific targeted NGS for noroviruses, where you just amplify the complete genome. That system is rather sensitive, I would say, to get a complete genomes, and I think the field is moving more than submitting only small sequences. I think it's going more and more towards you need to show a full genome of a new virus, for example, for your colleagues to agree that it is something completely new and not a partial sequence. So these assays we have been using in the surveillance platforms, and there are a couple of them listed here. For outbreaks, we have CaliciNet for 10 years now. Basically, it's the surveillance networks look at norovirus outbreaks at the state level, the state, county, or public health, the smaller labs in some of those states, and doing the testing and the typing and then uploading to a central database. They all have harmonized protocols. They use the same protocols, so there's no variation in sensitivity between the assays, so it makes it a nice way of looking at these viruses nationally. Then we have an EPI system, the NORS, Epidemiological Surveillance System. It's for all enteric diseases. Information is entered by epidemiologists, and that's primarily the disease outcomes and the exposure and transmission mode. Some of that information also entered in CaliciNet, but most of the disease outcomes is gathered from NORS, and we combine those two systems internally, so we can pull the data from CaliciNet into NORS and vice versa to have a good overview of the disease burden and what strains are associated with that. Then we have endemic disease. I don't have enough time to talk about much of them, but I would like to mention them. But in particular, the NVSN, the New Vaccine Surveillance Network, has been there for 13 years. It was initiated in 3 different hospital laboratories, 3 different states, before the rotavirus vaccine was rolled out. And that is a system, so we have all the data going back, and you see, using that system, we can see, and I'll... Oh, there's far. I was hoping it was the next slide. Sorry. Using that system will be later. Later in the presentation, we now see that norovirus is the most important cause of acute gastroenteritis, causing about 18-20% of all the viral diarrhea in children. Rotavirus is very low. We see we still see it circulating, but it's relatively low. And thanks to this system, we were able to basically follow that success of the rotavirus vaccines. And then we also look in the adult population. We work with Veteran Administration laboratories, 3 or 4, and we also do some community studies. So for CaliciNet, we have 30 different states that are participating. They're all certified, and they get PT panels every other year to make sure that the methods that they are all using are still performing accurately. And the states here in blue, we've tried to cover that with outbreak support centers. Those are the blue stars. You see them, for example, in California, in Idaho, Wisconsin, New York, and Tennessee, and then at CDC. And that way we get data from those states that don't have the resources or just have, for example, you don't have a lot of people living in North Dakota, so there are very few outbreaks. So if they have an outbreak, then they will send it to us, and we will further analyze it and type it. So that gives us a nice national picture. So the data are uploaded to the website, CDC website, so you can follow that. We update it every month. So if people are interested in what strains are circulating or in which state most of these outbreaks occur, you can go to a website, and like I said, it's up to date. So we have 10 years of data, so it's a lot of information. Now we can clearly see where the burden is highest. It's not really a surprise if you live in the US or in Europe, because long-term care facilities are those where a lot of the risk factor is very high once it's introduced. And the often subpar hygienic circumstances facilitates the virus to circulate in these facilities. You see more than 60% of the outbreaks can be found there. But you can also see the restaurants listed there, so that is often related to foodborne outbreaks. So that's a way where we could intervene by working with FDA and coming up with better recommendations for example, for sick leave for people working in restaurants. That is quite normal here in Europe, I know, but in the US that is a little bit different. On the right, you see the transmission routes and the person to person, and you see in gray there's still significant here, 14% of the total numbers, and if you do then the unknown, it's about 10 to 12% of all the outbreaks. There is a food is associated. It doesn't mean that the virus is detected in food, but it's a clear odds ratio. The outbreak has been investigated, and it's a clear odds ratio to a food item that a group of people had during the same time frame in the same setting, and they all had positive for norovirus, and then the same sequence, so matching. So what we have tried in the last couple of years is also to see if we can do some enhanced, more real-time, because usually the submission of all this information, CaliciNet was lagged, sometimes up to a couple of months. So we didn't have really real-time information. So we asked some of the states, were listed here, to do that more in real time, and that has been very successful. That we now have almost within 2 weeks, we have from those states, we have the latest information of the outbreaks. If it's foodborne, what types are circulating. So if a new type would circulating, this is a system where we initially would pick it up immediately. You clearly can see that the virus is, at least in the States, is very seasonal, with most of the outbreaks occurring between Thanksgiving, that's the last Thursday of November, and early May, so primarily in the winter, but you also can see that in the summer that is never really zero, so there are still viruses circulating in the summer months. interestingly, a lot of these viruses are often the genogroup 1. So the genogroup 1 viruses don't have a seasonality, while the genogroup 2, and particularly the genogroup G24, have a clear seasonality. So there's a different epidemiology between those genogroups. This is 4 years of data. Look at the blue, and you can see, by just looking at it without, it's a very busy slide, but about 50 to 60% is this G24 virus. So that has been very consistent. There are some other viruses have been trying to take over, and new viruses that emerged in East Asia, for example. We've seen them, but they never make up more than 10 to 20%. Whereas in East Asia, sometimes they predominated and caused almost all of those outbreaks for a couple of years. So there's still some differences between viruses infecting people in the US, for example, in Europe and Australia and Japan, and in other countries. So combining Calicinet with NORS, that's now gives us a really nice information as far as for where these particular genotypes are associated with. And you can see here in blue, the blue bars are the genogroup 2,4, and the other one are the non-G24. We made a distinction between those two, and you can see that, for example, foodborne viruses have a higher rate for these non-G24s. And so if you find these viruses and you have no information otherwise, like no epi information, you have a higher chance that you have to look for a foodborne exposure, and hence the intervention is different than for the person-to-person, as far as for trying to make sure that these outbreaks stay small-scaled and stopped as quickly as possible. So this is the slide I was just looking for. This is in the bar graph. You can see hospitalizations, emergency departments, and outpatient visits, and the total clinical visits. In blue, you see rotavirus, and in red, you see norovirus. And you can see that in the outpatient visits, as most clearly, and this was 2009, that you see that noroviruses are causing more infections than rotavirus. So that was the first years that we saw that, about 3 years after the introduction of rotavirus vaccines in the US. So this is a supernova. We love our acronyms. And this is in the veteran affairs hospitals. There's active surveillance. And so that's another platform where we get data about our different strains circulating and what is the burden of disease. The military is really interested in the US in a norovirus vaccine, both for active duty as well as for people that are retired and are often sick. And so that's another reason that we thought it would be a nice platform here to get some data. And this is some information that's coming from that. But we decided to use not only norovirus testing, we used one of those multi-enteric pathogen platforms. And you can see here that C. diff is the most important, C. diff and EPAC, but then as number 3 is norovirus, and here you see all your favorite bugs here, rotavirus, salmonella, STEC. But you can see that norovirus is also in this population one of the most important causes of AGE, acute gastroenteritis symptoms. So, but now we have the data for the US, and we had initially some data from MVSN networks, together with our colleague who was also here from Kansas City. We looked at some of the MVSN samples and looked at the types that are circulating there and compared it with the outbreak type. I'll show you that in a minute. One of the other reasons that we thought global surveillance is necessary, and I mentioned that earlier, is some of the countries listed here in red, in particular in provinces in China, but also in Korea and also in Japan. We had several years, we had new viruses coming up, and that basically the question was, okay, we have seen this G24, it has run its course, everybody is protected across protective antibodies, and now new strains come in. They did in these countries, and I said in the US we saw it, about 20%, but not all of them, and now these viruses are back to the background. But so we thought, well, having a global system obviously will alert us more quicker. And here's the slide I was just referring to, where we have some preliminary data that is in the same, more or less the same time frame, although I think there's a typo in the dates of the slide, is that, no, it is not, sorry. That this G217 here that was causing in that particular time period most of the outbreaks in Asia caused 10% of the outbreak. Here, another one in 2014, you can see this, it's barely 20%. And still in blue, this G24 boy is still there. So pediatric, you see here in the central in the pie, you see in green, this was another indication that maybe there are different viruses circulating in young children, because you see the sliver of G24 is in blue here is very small compared to what you see in outbreaks. So we thought, and also some study from Dr. Chan in Hong Kong that showed that some of those genotypes target different age groups, or at least there was an indication and trend that different strains causing associate with infect different age groups. So we thought, well, we need to set up some global pediatric surveillance network, focusing primarily on the pediatric endemic cases. There is NeuroNet, coordinated by RIVM and Rotterdam University. But this is focusing primarily on those children and see if different strains are circulating. So we have been working on that with the same principle as I explained with CaliciNet. We are now in phase 1. We have up to 10 different countries are participating now, and they're starting to upload to a web portal. And here's some data, aggregate data, in blue, again, the G24, but there are some other strains there that in certain populations, again, the G23s in these children, you see it is, but also the G22. So we have a better idea of which strains are circulating, and also if there are differences. Again, for a potential vaccine formulation, you would like to protect against the circulating viruses. And this is how it all will be visible, visualized with a world map, which we probably will have available in the next couple of weeks, where you can zoom in on the individual countries and look at the prevalence of the different genotypes. And this is in the US, an example, and this is, I think this is Hong Kong, the data from Dr. Chan's lab. And this is Germany, hospital in Leipzig there currently. So if there are hospitals here doing norovirus diagnostics and typing and would like to participate, please contact me after my presentation. So the cell culture, I want to move over this because Dr. Goodfellow talked about this yesterday. I think the major discovery there, as for most of the stem cell research, is basically the Hubrecht laboratory in Hans Clevers, in collaboration with Sato from Japan, to discover which of those factors are necessary to keep those stem cells alive. And that is being used by also for a system, the mini-gut culture system for these enteroids, where you isolate those stem cells and culture them first in 3Ds, and then for norovirus you need to make monolayers. So instead of rotavirus and other viruses that are able to infect the 3D, the organoids, for norovirus you need to make monolayer to get infection. So it's about 2 weeks, as is listed here, the time. So the enteroids, the 3Ds, the monolayers and the differentiation, and then the infection up to 3 days. And then you measure the differences by increase of viral RNA. It's a very expensive method because these factors are expensive. You can make them yourself by culturing 3 cell lines and let in the supernatant and purify those factors and use them, or you can buy it. we decided we buy it because it's a lot of work to keep these other cell lines running. But it makes it a very expensive technique. But it worked for norovirus. This is the first major breakthrough in 2016 that in Baylor College of Medicine, Mary Estes showed replication. You can see here in the bars, it's the difference, how you measure this between the black bar, this is 1 hour, and then here after 3 hours. So this is what you measure. In this case is the fold increase. You see here the numbers here. And you can see they were able to show the virus, they were able to show where the virus infected in enterocytes, and they were able to show that they were able to, although at a lower level, to able to passage the virus. I have to say right away, we haven't been able to replicate that, the passaging, and that's still one of the major challenging of this method, because we still have to go back to the stool to do these experiments. But here is showing some of those samples that we were able to show successfully that it also worked in our lab. There was, as Dr. Goodfellow mentioned yesterday, there have been quite a few of these new methods have been published by one group, and then nobody was able to replicate that. So we immediately, when this paper in Science came out in 2016, we worked with Dr. Estes and make sure that we used the same method and were able to implement this method successfully. So both G24 virus in the top, but also non-G24 viruses were able to replicate. No G1s or G4s, in case you are going to ask that question, already answered now. And also that it's not very easy. It's not every sample you can grow. Even if you have a high titer, a low CT, let's say a CT below the 20, the success is not guaranteed. Our success rate is about 20%, and that's shown here in this graph. So we have a lot of yeses here in the top, but you see in the lower, the 49, they also had high titers, but they were not able to see any replication. We still don't know exactly why, but that's at least something that we're looking into. Well, we use those stool samples that are successful for studies, like looking at chlorine. We all know that chlorine probably works very well based on outbreak investigations where chlorine is basically used, but we didn't have really the data to show that experimentally. So that's the first experiment that we did, and we found out that in a clean system as this is, so no organic loads except for maybe some serum that we, calf serum that we added, the up to as low as 50 ppm was able to kill this virus. So that's good. So we were able to show that chlorine worked. We also were able to show that ethanol or alcohol, like in alcohol-based sanitizers, hand sanitizers, may work for bacteria, but it doesn't seem to work for noroviruses. And that's very important information, of course, for sanitation when you want to reduce the spread. And you assume that these hand sanitizers alone will be able to reduce the transmission. That doesn't seem to be the case, at least on the initial data that we have. So the challenges of this Android culture system are listed here. Knocking down the innate immunity, we also have done it. And again, that is probably the best way to get a higher increase in fold increase. You have more than 2 log. You can do more studies in looking at inactivation of sanitizers. We use it for neutralization. So we now looking at antibodies, in particular monoclonal antibodies, and trying to do epitope mapping, looking which epitopes are neutralizing. So it is really a very nice system, at least as a start. But we really would like to have a virus that we can grow and culture and passage, and then maybe submit to ATCC. Then anybody who would like to do norovirus research can then order it from CDC. So then finally, I'd like to go over what the status of the norovirus vaccine is. I've told you about the disease burden, the enormous disease burden, the risk groups I've mentioned, the natural history of disease. Some of people can shed the virus for up to 4 weeks or sometimes even longer. Certainly when you're immune compromised, you can shed the virus for maybe up to a year. I've told you about the virus strains, which are the most important, G24. Most vaccine manufacturers are including the G24 in their formulation, and together with the genogroup 1 representative. But as I also showed, there maybe we don't know what the cross protection is. So that's one of the missing links here. We don't know if these two components are able to protect you against all these 26 or more different genotypes. And also the pediatric viruses, are they different? Do they need to have a different formulation? These virus-like particles are able to do that. The way how they are created is in different systems, E. coli, but also in new systems like here for the Vaxart. There are two companies that are in clinical trials. It's Takeda here, there's the phase 2. They finalized phase 2 and now going to phase 3 in children. I've been told, I haven't seen the publication of the phase 2 data, but the fact that they are moving to phase 3 tells you that they're investing a lot of money, so there must be a lot of support, at least on the data. And Vaxart is an interesting one. It's a company that's a small startup in the Bay Area, San Francisco. They make an oral pill, and they're working on flu and other viruses, but they also have a line for norovirus. And so I always say, well, I can take a pill when it's around Thanksgiving, and they say that they can reformulate the formulation very quickly. So we're excited at least to have maybe something in the nearby future that we can take orally. Here are some of the papers. So this is Takeda vaccines, and this is this Vaxart concept. It's an adenovirus, non-replicating, where you clone your second operating frame, and then in the gut it will raise immune responses. And it seems to be safe and immunogenic, so we're waiting on, and of course, tablets are very stable. If we want like to give this without a cold chain, that's possible. Not for children, where children and a tablet is a little bit difficult. You need to come up with a liquid dosing, but for this this stability aspect, I think is very important for a vaccine. So we'll see. So the challenges already mentioned. So which genotypes do you need to include? Does a potential vaccine need to be updated regularly? Is there protection against multiple types? And quite a few are circulating, as we see a lot of trends over the years. Is there a role of prior infection history? You think about the pediatric vaccine. Think about antigenic seniority, as we see with some other infectious diseases. We don't know that yet. Duration of protection. Do we need to have one shot? Probably not. Lifetime risk for all of us here is about, on average, 5 norovirus infections. They may not all be symptomatic. Some of them may be asymptomatic. And of course, most important is the cost benefit. And there's now one study is done, and I think more are in the pipeline to see in which markets a vaccine will be cost benefit. So in conclusion, the pool prevalence of norovirus, as far as we know from 175 studies, is about 18%. Outbreaks in the US, mostly in adults. In China, mostly in schools, or so in the younger age groups. Long-term care facilities, they don't have a lot of outbreaks that are occurring there, or there's not necessarily a reporting requirement for those. We see them in outbreaks and endemic. We see the different strains. We see some differences in the US between children and what we see in these outbreaks. So we'd like to monitor at least for a couple of years, see global trends, different strains circling in different continents. I showed you about the non-transformed human intestinal enteroids, or the mini-gut system, that is now implemented successfully in several different laboratories, including in Europe. So I'm hopeful that will give us some more information about targeted interventions, for example, antivirals, and maybe vaccines. And we'll see that it usually takes a long time, and we'll hopefully hear in the next half year what the Takeda vaccine. Why did the company decided to move to a phase 3 trial? With that, I would like to thank at least all the people in my lab who did all the work. I'm only presenting here, and all the collaborators on the right side, I'm not going to mention it. Well, I'm going to mention a couple of them because they're in the room. Raj Shelvaniara, Kansas City, he's here. He gave a presentation here. And Martin Chan, he will give a presentation later on, not about norovirus. Shame on you, Martin. And of course, we work closely together with our EPI team at CDC. So thank you very much. Happy to answer questions.